All right, this is a very interesting article for everybody that's been focused on climate change and specifically methane reduction, because methane is supposedly 50 or 70 times more destructive in the atmosphere than carbon dioxide. It's a greenhouse gas in a very big way. So let's jump into the article, and then it's a Q&A. It's an interview, and it's very informative. So let's just jump in, and then we'll give some follow-up comments after the end of the article. So here we go. The title of the article is CRISPR, a game changer for livestock methane reduction? Question mark. It's high risk, high reward, says UC Davis professor. So June 26, 2024, Elaine Watson. And here we go. Article begins. From feed supplements to animal breeding to vaccines, multiple approaches are being explored to cut methane emissions from belching ruminants. But if you can edit the genes of the microbes responsible for the problem with a one-time treatment, says Professor Ermias Kebreab, you could change the game. It's high risk, high reward, but if it works, you won't have to do anything else. A potent greenhouse gas, methane is generated by manure as it decomposes. It is also burped out by ruminants who produce it in a stomach compartment called the rumen. Here, microbes break down complex carbs and produce gases that are converted into methane by another set of microbes, methanogens. Dr. Kebreab, a professor at the University of California, Davis, who has conducted extensive research on livestock methane reduction, is one of a team of academics working on a $7 million initiative backed by the TED Audacious Project. Led by Dr. Jill Banfield and Dr. Jennifer Doudna at UC Berkeley, pioneers in metagenomics and genome editing respectively, the 7-year Engineering the Microbiome with CRISPR to Improve Our Climate and Health project aims to develop a precision microbiome editing to address two challenges associated with microbes, childhood asthma and livestock methane emissions. So AgFunder has caught up with Professor Kebreab to discuss the livestock methane component of the study and other approaches to dealing with cow burps. So first question, in a nutshell, what is the CRISPR project looking at? The response, our research on Asparagopsis red seaweed used as a feed supplement to inhibit methane production via interfering with enzymes microbes need to make methane shows that the decrease in methane wasn't caused by a decline in the methanogen populations, the microbes that make methane, but by downregulation of genes responsible for methane production, which identified a mechanism we could potentially target in the CRISPR project. In the cow's rumen, bacteria first break down the cellulose and other material in cattle feed and produce hydrogen and carbon dioxide, which methanogens then use, producing methane as a byproduct. If you can identify the genes in these microbes that are responsible for methagenesis, it may be possible to edit them out so that they would otherwise function and still use hydrogen as a source of energy but won't produce methane. Next question. How might it work in practice? Response. CRISPR is a pretty new tool when it comes to microbes as opposed to plants and animals. The idea is to find a carrier that will take the CRISPR CAS system to specific microbes and then edit it. We also have to figure out how to package it so we can give it to calves via an oral supplement before the rumen is fully developed, so we'd be effectively reprogramming the rumen. It's a high risk and high reward project and definitely one of the most exciting approaches to enteric methane reduction, as if it works, you won't have anything else. You won't have to do anything else. Our editing system could target several of the microbes that make up the cow's microbiome, communities of microbes in the cow gut. So the first step will be bringing the microbes into the lab and characterizing model versions of the microbiomes essential to methane production. The next step is to develop the tools so we can deliver editing tools into the target organisms individually. Next question. How well do we understand the microbiome of cows? Will you need to edit several different kinds of microbes for this to work? The response. We have started some research starting from calves all the way to adult animals, and we are taking samples every month and trying to understand the development of the microbiome throughout their lives and trying to identify the microbes that are important and needed to make the rumen stable. Then we need to understand what happens to the animal if you inhibit methanogenesis, so we have experiments going on to try to understand that as we want to have a functional microbiome, we don't want to impact microbes that help maintain this microbiome. Next question. Do you already have some insight from your trials with seaweed supplementation into what happens in the rumen when methanogenesis is inhibited. There's only one long-term study that was done in dairy that we have to go by, but we don't really know from that the makeup of the microbiome. So we're looking at studies now where we try feeding calves seaweed until 3 weeks after weaning, and then we stop and see if there's any kind of reprogramming that we can do, just giving them this additive at a very early age. And then there's another treatment within the study where we look at continuously providing seaweed supplements throughout the cow's lives and see what it does to them. Next question. Can you share a bit more detail on who was doing what in the CRISPR project? A response. We work with UC Berkeley and UC San Francisco, and our role at UC Davis is focused on the livestock part, where UC Berkeley is focused on the CRISPR tech itself. So at the moment, we're collecting samples of microbes from cattle rumen and getting the DNA from the organisms that we send to Berkeley. Finally, if and when we have a system figured out, we'll do the animal trials of the final product, likely an oral supplement that can be given to calves to edit microbes in their rumen at UC Davis, where we have our own dairy farm. Next question. What progress has been made so far? Response. Over the past 6 months, we've collected a lot of samples from a trial that is right now with 5 calves, but we are aiming to get up to 24 by the end of the year. We're taking samples every month from those calves and analyzing what's happening in the rumen. Next question. Stepping back, where do you see the CRISPR approach fitting into the broader toolbox of intervention when it comes to enteric methane reduction? Response. We have to come at this problem from multiple different directions. One approach is to breed animals that are inherently low methane emitters, and that's happening in New Zealand with sheep, where they have sheep that are about 12 to 20% lower in methane production. That work is being done in cattle as well, where the natural variability in terms of emissions is about 30%. So that's definitely one of the interesting tools in the toolbox, especially for grazing animals. The drawback of this selective breeding approach is that it will take a long time. Next question. Can we learn anything from animals that naturally emit less methane that might help inform other enteric methane reduction strategies? Response. We don't know a lot about why some sheep produce less methane than others. It could be that they are more efficient at feed conversion, or it's something about the makeup of their microbiome, but we don't really know too much about it. We need to find out exactly what makes an animal inherently lower methane emitting. Next question. What is the potential of seaweed, Asparagopsis, as a feed additive for methane reduction? You can see huge reductions in emissions from seaweed, but there's still more work that needs to be done, and which is being done to ensure it's safe for animals and people. So we don't have an issue in terms of the increased level of iodine and bromide. It's less of an issue in beef cattle because these substances don't really accumulate in the cells, whereas in dairy it could impact the milk. Although you can grow seaweed with less iodine. With bromide, it's more challenging because you want bromoform, one of the key components in red seaweed that inhibits methane production. So how do you make sure you have enough bromoform, but you don't have bromide in the end product? Next question. You've also done some recent research on grape pomace as a methane inhibitor. Response. Grape pomace, a byproduct of wine production, contains some tannins and lipids which have been shown to reduce emissions. So we did an experiment looking at if we supplement with an appreciable amount of grape pomace, 10 to 50% of an alfalfa-based diet for dairy cows, what impact would that have? We saw about a 10 to 12% reduction in emissions, better feed efficiency, and a positive change in the fatty acid composition of the milk. And final question, what in your view will drive update of feed additives or other tech to tackle enteric methane production? Response. There has to be some sort of incentive, so you'll probably see more big players that buy a lot of milk, generating carbon credits or assessing funds that will help cover the cost of feed additives for farmers. But I don't see many farmers doing it themselves right now, whereas the multinational companies have already committed to reduce emissions. Okay, so end of article. And here's our 2 cents for being investors in seaweed. Asparagopsis is really a tough seaweed to grow. It grows about 1 to 2 meters below the surface of the ocean. It usually needs to be fixed on ropes or natural rocks. It's extremely difficult to produce. I think CH4 in Adelaide, Australia, is one of the few companies that say they are going to do it, but it seems from other folks that I'm talking to in the seaweed growing industry that a lot of these companies, specifically um, the aforementioned, cannot really yield or produce a whole ton or however much is needed. But the other part about methane reduction using Asparagopsis is the fact that um, it's not economically viable yet. You have to really force farmers to buy something that's technically an additive, a few grams for a larger quantity of grass. So if there's not a subsidy or some government incentive, the farmers are just going to see it as a higher cost. And then one final thing that I heard from a conference that I attended recently was that supposedly, specifically down in Australia, when the butchers are butchering a cow that has been fed with Asparagopsis, the Asparagopsis is potentially or perhaps doing some weird things to the cows. So that's our 2 cents from the natural Asparagopsis side. Back to this article. This is quite interesting because it seems that this professor, Dr. Kebreab at UC Davis, California, um, is trying to do the trial and error with young cows to basically gear the baby cow's stomach and sort of plant this bio um, this okay the bio yeah the stomach will be able to have a new flora, and that's really the key to that approach. Uh, so that you're basically teaching the cow from a very young age that you're replacing whatever it's supposed to be growing up with right after it is weaned from its mother's milk, and then coming in very fast for the microbiome, the flora of the cow's stomachs to be geared towards the Asparagopsis' effects, uh, all of the positive effects that are sought by giving Asparagopsis to cows in order to reduce methane production, et cetera, et cetera. It seems with CRISPR um, that this is a DNA-focused project. So as a whole, these $70 million that are being that was granted, it seems, are split across 3 different state universities in California: San Francisco, Berkeley, and Davis. Um, and up in Berkeley, they're dealing with CRISPR itself. So Kebreab is dealing more on, it says, the uh, cattle part, the livestock methane emissions. Um, so this article speaks really more to the cow part versus what CRISPR is. Um, so yeah, CRISPR is very high tech, but the cow stomachs is something they got 4 of them. Um, they're the Asparagopsis is it's tough to farm. Asparagopsis is hard to get the yield. It's prohibitive for farmers unless they're subsidized by the government, and it might screw up the cow stomach or screw up other parts of the cow when it comes time to slaughter. So great article, very interesting. But Asparagopsis overall seems to be, the jury's still out on what's going on there. And that's our 2 cents from what we have heard, and that's where we will leave it for now. Wonderful article, really appreciate it. Good to share it with you all as well and offer our commentary. And looking forward to covering other articles that deal with Asparagopsis specifically, methane reduction, and similar activities.